while you are making that observations chart, I'm just going to quickly remind you about a couple of things here that'll be going on when we're in the lab. Okay, up front on the main kind of teacher's desk up front there, okay, are all of the unknown samples. They each have one dispensing apparatus inside. By dispensing apparatus, I mean for the solids, there's a scoop. Okay, for the liquids, there is a eyedropper. That thing, yeah, you know, that you squeeze and stuff goes up in an eyedropper. Okay, and then those need to stay in the container that they are in. If you use one scoop from one in another, you'll contaminate the entire sample. I have 3 classes that need to use this. Mr. Hesh has 2 classes that need to use this. So we would prefer not having to throw away large amounts of sample because you decided you needed the spoon from A for B. Okay, so just make sure that you are not using it in more than one container. Keep it in the container. All right. All of the other lab equipment that you require is on a silver cart at the front of the room. Okay, on that cart are the depression plates, the conductivity testers, and the pH paper and the thermometers. Okay, so I would suggest, so that the front of the room does not become a total gong show, okay, that one person in your group is responsible for going and getting the materials. Okay, one person only. Right? They go up front, they grab all the materials, come back to the desk, and then that same person brings the depression plate up front to put all of the unknowns in the depression plate. What should you do to the depression plate before you bring it up to put the unknown samples in it? Yes, I would first wash and dry it, okay, just to make sure that it is perfectly clean, and then I will come around with markers for each table, and you should label it. Okay, label each depression A, B, C, D, so that you put everything in the right depression and you know what sample is in what. All right. Now, there's one sample. A note for sample C. Okay, for sample C. It is already dissolved in water. Okay, sample C has already been dissolved in water. So, for your observable properties for sample C, okay, you should write in, okay, that sample C was a white, crystalline, solid, with no odor, and small trapezoidal crystals. Okay, just because it's already been dissolved in water for you, okay, so that you know, when I dissolved it in water, it dissolved easily. So you might want to write that for the solubility part, okay? Okay, so it dissolved easily, and the solution got slightly warmer, like there was like a 1 or 2 degree increase. It was nothing major. Okay, there was a slight temperature increase. Okay, and one other little tip for you. We talked about this the other day. One of your unknown samples is ammonium nitrate. Okay, one of them is this stuff. Okay, it is made up of 2 polyatomic ions. So what kind of a compound is it? It's ionic. Okay, so keep that in mind. I talked about it last week, and I said that it was used in something. Does anyone remember? Right, in the instant cold packs, the ones that you pop. Okay, so when you pop them and it mixes with the water that's in the bubble, it gets colder. Okay, one of your unknowns is that stuff, something you'll want to watch for. Okay. I think that's all the stuff I needed to tell you. The rest of the stuff will work exactly as we had planned. Okay, remember that the tests you are doing, you will start with the observable properties test. Okay, record everything that you can see about that material. Okay, do the wafting test of the main samples up front, just one person, okay, someone who isn't congested and can smell, okay, and then do the solubility test, then do the conductivity test, then do the pH test. Guys, we do want to try and save resources here, so you don't need very much pH paper for the pH test, okay? Really, a centimeter long is lots, okay? Everybody follow there. The pH papers are in plastic containers, okay, and the little color code is inside the plastic container. Take the color code chart out of the plastic container if you get one of the orange colored plastic containers, because if you try and look through the orange, it alters the color of the chart. Okay, so take the color chart out when you're comparing colors. Okay, any questions before we head across the hall? Okay, one last thing in terms of lab safety. I need your hair tied back if you have long hair. If you're wearing a hoodie or something with drawstrings, please make sure that those are tucked away so that they don't fall in anything. Okay, if you do not have your feet covered with proper footwear, you need to go and get some proper shoes, or you need to stay away from the lab bench. Okay, if you're wearing any loose clothing at all, you know, just make sure that it's either leave the loose clothing in here if you can. Obviously, dress code still applies. Okay. Otherwise, just be mindful. Okay. We want to stay safe while we're in the lab. Any questions? Okay. Okay. So what's left for you to do now that you have all the observations, is to take those observations and compare them to what you think the properties of these various unknowns should be. Okay. If you want, of course, you can look them up. You can Google them and see what their properties should be, but I don't think really you're going to need to do that. Okay? I think that from the observations you have, you will probably just be able to identify them. You might have one or two at the end you're not sure about, then go ahead. By all means, you can Google those. That's not cheating. Okay? That's just using resources that are available to you. Okay? So what I want to see then is for your, all you have to do for your analysis is fill that chart in. Okay? Have them identified and tell me whether they're ionic or molecular, okay? And then you got to write your conclusion. So again, copy and paste your hypothesis and tell me why what you did in the lab supports or rejects it, okay? Your evaluation, 2 sources of error and how to fix them. You do not need to do the class discussion questions, okay? We will talk about those in class on Friday when I am returning your labs and talking about it then. Okay, so it's due on Thursday. If you want me to look it over, it's got to be done ahead of time for either tomorrow or Wednesday. Okay, then I can look it over and schedule helper at lunch. Austin, question? I have the results already posted for you. So if you're wanting to double check your data, my data is posted on Google Classroom in there. And so there'll be 3 podcasts, your file that you're working on, this file, and that file that with the tests that I posted Friday, and then the results. So there's a lot of stuff that's in that task right now. It doesn't necessarily mean you need all of it, but you should watch the podcasts while you're putting it together, okay, just to make sure you've got everything there. All right? Any questions on that? Okay, you still have a little bit of class time, so I would use it effectively, okay, like 2 or 3 minutes here.